Hello interstellar everyone, welcome to class 11 biology. With this video we are going to start a new chapter from our NCERT textbook, that is chapter number 10, cell cycle and cell division. In our previous video we studied about cell, the fundamental unit of life. The cell as a fundamental unit of life. And then we studied about cell theory. And the cell theory Rudolf Virchow ki modification omnicellular cellular laro. Omnicellular cellular lan khalo gore a new cell arises from a pre-existing cell. The khalo gore cell sawachik cell nyama ningba yogyo ni chago reslar. Ta thanda ngazu chapter dina ngazu kharjangare the khandes yungu yumedi. Cell ningba jene cell sawa khandes tukku yume. Or we can say how a pre-existing cell creates a copy of itself. Pre-existing cell chigirwa. How does it increases in number? Ko khandes phege to gore. Ko mangro khandes to gore. Ko gi cell sawa khandes tungore. Igana jangir chapter dina ngoba. And you dordu rushe we manner chapter dina chasan rusta we manner kharugam jangir we manner. There are two types of cell division. Cell division come under near. Mitosis ta meiosis la near. Mitosis and meiosis. Dini kyanzu yabu gotsyogu gorda. In biology it is very important you understand the two types of cell divisions. They are mitosis and meiosis. Angazu thangbu ngazu khargan jangir lawi bena ani cell cycle lai dikan jangir. The cell cycle go mansu nye ne kyanzu kharu gotsyogu gore lawi bena. Organism khande mo khande imbaine. Whatever type of organism you take, unicellular imbaine re, multicellular ngazu doji imbaine re, ani zamni nanglo ne sokja cheshu shingdong doji imbaine re, blue whale doji imbaine re. Onzo thangbu chadu sambala. They all originate from a single cell called as zygote. All sexually reproducing organisms, they start from a single cell called as zygote. We are just a single cell. Amegi konga di power gi sperm gi fertilize cheni ani thangbo cell chik chagro cell chik dina ngazu khala wore zygote re. A cell chik dine ani ngazu ta thanda ngazu de we ngazu subu de form che ro, which is made up of trillions of cells. Ngazu subu nanglo cell pe mangu yor. We all have formed from a single cell called as zygote, and that zygote it divided again and again to create a multicellular organism like us. Di imba sonza ani cell cycle, the cell division kan changor ta ngazu. Let's see what do we mean by cell cycle. Cell cycle ki definition kare. What is the definition of cell cycle? The definition of cell cycle is the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell itself, and eventually divides into two daughter cells. That is called as cell cycle. A cell cycle la zoma khalo ore cell chigne cell ni la ma macha nye ne ro. I para la khar chungor events mangbo shu chungor khar events khar chungor la mena cell nye la ne sha yu chumare din nanglo yo ki genome la mena DNA the RNA dige sang duplicate chi na khalo ore. They create extra copies of itself. Ross kya wana shi khaji ore ko copy theos yo DNA da nezo. And did the nyamdo la khaji ore other constituents of the cell like cell organelles, proteins, biomolecules shimpa dige sang khaji ore by synthesizing them, and eventually it divides into two daughter cells. Sadi mangro tango yumze kharla na cell chik nanglo la khar yo wadi sangma cell ni la pha kopsha kya go warwa di du sambala onzu nanglo yo kyo genome the constituent sangma khaji ore divide chart war or khaji duplicate chart war extra copy create chi war so chung so the sequence dige sangma la nganzu khala war cell cycle la war so chung so. Now the cell cycle, it consists of two phases. Cell cycle la jo marwe ila khare phase near rimba near phase ni dikhare la women interphase ta M-phase. Interphase is also called as preparatory phase ngal detail na jangar da, and then interphase is followed by M-phase la na mitosis phase la or so chung so interphase and M-phase tadi kesi cell cycle the remote on the way mena kira lotem na remote da e remote shambas tum na lol. So let's say this is a cell cycle. This cell cycle chirda cell cycle nanglo la gozu sa dine doji nimba chacha wa cell chik ne gozu wa chacha cell chik the cell cycle gangsa kora chik gap sa vechela it will form two cells, two daughter cells. Now I don't know why it is called as daughter cells, why not it is called son cells, but it is called as daughter cells, right? You may come up with the reason these two daughter cells they will become mother cells later on. They will divide again and they will create more daughter cells. Okay, so these here, so they are called as daughter cells. Ta daughter cell di dinang la ko chigne ni chaso ni cha lo ko rimba mo shu do do rimba mangbu di nganzu gi ta ngana stop de wo me che do nganzu gi face major face ni la ma design ni le yawa che shar har gar re la win ba na ine ngombu rimbu shtong do ta rimbu sto ngombo 보고 싶으면 돼. 딜러 가지고 하려고 그래. Interphase. This is called as interphase. Interphase 사와단 가지게 돼. The cell will enter into mitotic phase, M-phase. M-phase 사와단 가지게 the cell will divide into two daughter cells through cytokinesis. 요청소. Interphase 체 근데네 M-phase. and M-phase 사와다 가지게 cytokinesis 게 through cell 니 하고 싶어 돼. I hope it is clear, right? Cell cycle lana kiranzu remove the inji jangorda. You have to practice the one given in the diagram which is given in your textbook. Textbook na te yoga diagram de abu practice chima do. Now we will dive deeper into interphase. Interphase ki kala detail nembe ne chayanda. Okay. Now interphase la we imbana khala wore. What do we mean by interphase? The dinecheni dipo do yokyo de ngazu khala wore. Interphase la wore. The cell is passing through interphase. The interphase is also called as the resting phase. Interphase la resting phase la because there is no actual division happening over here. Cell divide ke chigo marti nanglola. So it is also called as the resting phase. But don't be fooled by the name resting phase. Resting phase la di taniro ani cell bat kor relation de oru tsiye marta. Hajindana it is the most active phase of the cell cycle. Interphase is called as resting phase, but it is not resting. Actually, the cell is not resting. It is not dormant. It is very active. In fact, it is the most active phase of the cell cycle. Resting phase it is in fact called as preparatory phase. The cell is preparing itself to divide now. Interphase kab du cell koran divide the chigo mardine, it is preparing itself for division. Cell division the prepare chigo. Cell division the prepare chidu zambala ta khari chigo la wim baina. 여기 코로나로 여기다 자기 뭐부 주요 여기 genome 여기 나로 constituent 되게 상마 replicate 해야 되게 상마 interface 중구요가 송자 it is called as preparatory phase. 이 맛에 interface is the longest phase of cell cycle. Interface 라디 페링부어 interface 라디 투즈페링부어 it is in fact the cell spends most of its time in interface. cell 망체 가르치고래 투즈 망체 가르나던 거라고 하면은 interface 나오. The cell is preparing itself to divide, so it is spending most of its time in interphase. It is given here that 95% of the cell cycle's duration is composed of interphase. Cell cycle di chazanjik cell cycle chazanjik imaina di nanglogi gaja gubju gonga jomare ikare interphase chalu. Or you can say that isi cell chik ne cell ni chaya jomare cell chik ne cell ni chaya la chujuji gorgu yogi by na 95% of that 1 hour, let's say 57 minutes kamang abjung abdu interface na dedi gor cell di interface kamang abjung abdin sawada ani M phase di khajuma de yomara kama summa de yomar only 3 minutes out of 1 hour is in M phase, 57 minutes is in interphase. So 95% of cell cycles duration is spent in interphase. The interphase like the kechum burdo interphase like it is a preparatory phase. It is the most active phase of the cell cycle, and the cell spends most of its time in interphase. 95% of the time is spent in interphase. In the interphase like the design sum la yewa chigir interphase na lo activity mangu pe chungu yewa sonza ni ngajo gi interphase like the design sum na yewa chigir da design sum di khar khar rela na G1 phase, S phase and G2 phase sequence dakhra na larda G1 phase, S phase and G2 phase in this particular sequence, in this particular order. Okay, so let's see what happens during G1 phase, S phase and G2 phase. In G1 phase la dizambala khala or gap 1 phase la it is called as gap 1 phase or first growth phase. Kiranzo loktep shimba na la khar thongure la wemena first growth phase lanto, or you can call it as gap 1 phase. Okay, G1 la dizambala gap 1 phase. Di kabla khar chungore la wemena the cell grows in size. The cell chomara cell chik chora ta ko divide chela 30 chora it is amal ko khajire size chirdur. The cell always grows in size before it divides. Actually the growth is the reason why cell divides. cell grow mache way by na cell divide che go jumze cha go mar cell cher du zambalar ta cell cher du zambala kisi ko cell divide mache way by na ko unlimited chembu cha yo mar there is a reason behind that khajin na cell chembu shu cha way by na ta ko gi surface area ta ko gi nanglo volume jo mar disproportionate cha ru kuni ni para proportion jo mar disproportionate cha du zambala cell kora survive che tu go mar 그 surface area la ra ligro oxygen khaju ju ma ju khala khaju ya nanglo le male la dige changma chu khaju ya ma do ma do la dige changma cell gi surface area la ra ligro isi cell di chembu shu cha wi ba na volume mount surface area nyung tha din zabala cell survive che tu ko mar ani khaju gore cell di khati ne divide cha ro divide ma cha wi ma cell di share so during G1 phase which is called as the gap 1 phase the cell grows in size cell chirutur. And during that particular time, protein synthesis happens. Protein synthesis goes over. Protein due to enzymes. Most of the proteins they are enzymes, right? Enzymes re. And ATPs, RNAs, nucleotides they get sangma hajire synthesis goes over G1 phase la. The kharche do ready chegore S phase che do ready chegore. The cell is preparing itself for S phase, synthesis phase la ready chegore. So the khare mazu remu delta na tir. The khare G1 phase. G1 phase kabla cell grow chigre ko nalo liyo yo protein da nucleotides da dige sama chigre tege tour power tour synthesize ya go tour. And then next phase is S phase. Phase ni wa dikhare S phase. Interphase nalo ge phase ni wa dikhare S phase. S phase is also called as the synthesis phase. Di kabla cell grow chigo mana di kabla khar chigo la wi mana there is a replication of DNA. Tadi pe nenga chumburta. Chungi nalo la kena DNA replication kana jangro ati du sama kena space yo chalur thanda kena khar go chona di gore cell gi nalo nucleus nalo la ngajo khar yor laimbe DNA yor laimba in the form of chromosomes S phase kabla khar chi gore la na nucleus nalo lo yo gi DNA dige sama khaji gore double cha do the DNA amount doubles during this particular phase so during S phase the DNA replication occurs because of replication is nothing but the copying of DNA. Because of this replication of DNA, the amount of the DNA, it becomes double. Double amount. And then yamdol kharchir la wimbana during S phase, you see animal cell doj imbana. Centriole dash centriole and centrosomes lang. So animal cell na centriole ke khachire duplicate chir. Centriole ke dab maurdur chik modo me wimbana ho khachire ni liquid. So the S phase se uchungso. Because the main DNA copy called S phase because there is synthesis of or replication of DNA, therefore it is called as S phase or the synthesis phase. S phase G2 phase. G2 phase is also called as gap 2 phase. G1 like gap 1 lamba. G2 gap 2 phase. G2 phase there is protein synthesis and synthesis of other molecules necessary for the next phase. The G2 phase M phase G2 phase ki M phase re din zambala M phase kabdula mitosis ngone chungdu zambala protein mangu shuj ko or enzyme mangu shuj ko or dige sangma go yoba sonza ani G2 phase kabla dige sangma synthesize chir again the cell grows in size and in some textbooks you will find that G2 phase is also called as second growth phase so G1 S and G2 phase which is part of interphase. Interphase can be subdivided into three phases: G1, S, and G2 phases. It is called as G0 phase. G0 phase G0 phase G0 phase is also called as quiescent stage. Quiescent the dormant dose dormant active G0 phase especially the cells which does not divide. This is cell division divide cell cycle so G0 phase cell the cell which no longer needs to be divided. Cell which no longer needs to divide is they enter into G0 phase. They exit the cell cycle. Cell cycle le machilo thendure ani ko G0 phase na dur ani tene che divide chigmar ani chasha bane desia interphase nanglo la dikhare G1 phase ra cell G1 phase na ju ta kisi ko divide chego me wi mbana ko khachi re cell cycle ne ma chilo thön ru ran ko hol ju do la wi mbana G0 phase la ju so ena cha na nga ju so bu nang lo la heart cells choma it had they most of our hearts nga ju sang nga ju gi ning gi tabun jo ro cell they have entered into G0 phase they won't divide again therefore if our heart cells die they won't regenerate ko kone kharchi re yar get dich kharsa so ya yo mar so chung so azu gi skin cell do jim bai na khachi re regenerate che yo ro cell divide chan khachi re ma du kap du da kisi ning la ma do so che wo yin bai na ning is tabun ro bang bu shu shi wo yin bai na and then they cannot be regenerated therefore we will die so chung so heart cell they have entered into G0 phase that is so chung so G0 phase the interphase ki chashe marda it is just a special phase in which the cells that does not need to divide they will enter into G0 phase the cells which divides continuously they will keep on cycle in this particular cycle and then all interphase and M-phase la ni yorwa so chung so akira zo interphase la de se chung yir and next dikha re ngan zo gi M-phase ro the cell cycle gi interphase se chung so interphase na lo la phase sum do G1 S and G2 G2 to sawadan kharchi re cell will enter into M-phase M-phase is also called as mitosis phase or mitosis phase lag re it is the phase of actual division the kabla khaji bor shine the cell divide che ko chu bor it is the phase of actual division because interphase kabla cell divide chi me kharchi bor cell is preparing itself for division during M-phase this phase the cell is actually dividing therefore it is the phase of actual division the M-phase na lo la ya even ni yorta M-phase na lo even year tanji interphase na lo even sum yo na shi M-phase na la event near event need nenga chimburda even thangbo dikhare la wi mena karyokinesis which is nuclear division cell na lo nucleus yora thangbo khajire nucleus di divide char cell lamsa dokla ni le chag marda thangbo khajire cell na lo nucleus di divide chini ni le chagre nucleus ni le divide chazana ni shine khajire it is followed by cytokinesis which is called as cytoplasmic division so nuclear division dina ngazo khalegre karyokinesis nuclear division chaza jela ni chilo gi cell gi ani cytoplasm tha ani cell membrane gi du slow ga chadwe dela and khalo ore cytokinesis lower so M-phase is composed of two events they are karyokinesis and cytokinesis karyokinesis means nuclear division cytokinesis means cytoplasmic division and M-phase mitosis is also called as equational division ta kan thabu ngazo khargan jangla na mitosis kana jangarta mitosis is also called as equational division khajin lagore means cell chigne cell ni lama tudu zambala cell chig na lo chromosome khaju yo jo mare di chikpa chigyan cell ni lama ko shakir this cell chig na lo chromosome nga yo bi bana total cell ni le khaji cell chromosome nga ko shakir ta ngaju suburo jin bana ngaju subuna khaju re chromosome 23 pairs of chromosomes here ngaju cell gi ngaju subuna lo cell den zola there are 23 pairs of chromosomes and if our body cell undergoes cell division through mitosis ke through cell division chi yo bi bana cell chigne cell daughter cell ni form chi tuma daughter cell ni ka na lo khare there are 23 pairs of chung sa shi du ma te shir di ma song the mitosis la khala or it is called as equational division chung sa mitosis nang lo la ngazu stage ni do karyokinesis ta cytokinesis bu ngazu kharta gre karyokinesis ni khalas tangbu cell chigne ni ma cha nya tangbu haji nang lo la nucleus divide chi gola so ngazu tangbu kharta go ore karyokinesis this sequence ta do gorta interphase nang lo la G1 S G2 che and then M-phase nang lo la karyokinesis tangbu ta go dijel khare cytokinesis ta karyokinesis ta du zambala karyokinesis la di ya khachire stage shilama yawa cheshar karyokinesis occurs in 4 stages and remember this is done for convenience stage dige sama bath kora contrast chu yomarda dine dipado in dire la yomar because it is a continuous process karyokinesis nalola stage shiter the prophase metaphase anaphase telophase linear p met lan jana digir kiranzo p at P stands for prophase, M stands for metaphase, A stands for anaphase, T stands for telophase. We are just studying these 4 phases. as karyokinesis because of for our convenience. Maranzo jang deu yang chu chigo or mado shimba. Dine dipa do prophase reda, dine dipa do metaphase reda, dine dipa do anaphase reda lani. That koro dum cheyo and yo mar. Kachinani it is a continuous process. Cell now hachu gore. The process is going in a continuous manner. So manzo ki design na yewa che de maranzo jang deu yang chu, maranzo go chue deu yang chu chigo or mado. Ngoyo na ngolo la ro. Cell divide chi du zambala prophase. Dijalani metaphase and dijala anaphase, dijala telophase lani. Tumbu na yo marda. So karyokinesis it occurs in 4 stages. 4 stages the karre prophase, metaphase, anaphase and telophase. The sequence thing auto joel ni khala so mat. Mat lanandi. I mean you can come up with any kind of combination of words, right? Mat. That's all. Prophase. So we will go through them one by one. Prophase kab to kharchung gore. Metaphase kab to kharchung gore. Anaphase kab to kharchung gore. Telophase kab to kharchung gore. Meta gore. So prophase kab to kharchung gore la bhi na. Prophase na since we are talking about nuclear division, we will focus what is happening within the nucleus. Manju ki dikhare. Karyokinesis ki chashero. Karyokinesis la na khari nuclear division imba. So prophase, metaphase, anaphase, telophase changdu zambara mazu khar kanta digre nucleus nanglo lo yuga khar tu do ta digda nucleus constituent chi meta gar ta thambu tadu zambala prophase kab du khar chungore there is untangling and condensation of chromosomal material, that is chromatin. Interphase ki kabla ta the prophase machange ke jarda G1 S G2 into zambala cell gi nanglo lo nucleus yorwa nucleus nanglo la chromosome dige sama chromosome form na yo marta. They are present in the form of chromatin, thin thread like materials called as chromatin. Interphase ki nucleus shomarwe di nanglo la chromosome la ni thongu mar khachire nanglo lo kuba do ma be thongu dige sama khare chromatin re. Ani chromatin di khachigo re jela prophase na dudu zambala when the cell enters into prophase na judu zambala M-phase na juna thangbu khachire prophase na juro prophase na judu zambala anji kuba do ma butida the de yoro shme dige sama khachire they will get untangled kuba do ma buchi nanglo lo yobe man sama tida the yoro thongura prophase kab du khachigo re di tida the yo sama shigdur amewa cha. And then the chromatin starts to undergo condensation na ko chung dur. It will become thicker, but it will become smaller. Condensation of chromosomal. Chromatin will condense to form chromosome. Start forming chromosomes. Sorry. And the nucleus nucleus ki chilo centrioles will move towards the opposite poles of the cell and starts assembling spindle fibers. Animal cell doji by na chilo centrioles centrosome ki form and then they will go towards the different direction. Prophase kabla chromatin they will undergo condensation, right? Chromatin fibers they will undergo condensation to form chromosome kali ko zur prophase ki kabla da ani centriole chilo nucleus ki chilo nuclear membrane ki chilo centriole cytoplasm centriole digi khachire they will move towards the opposite pole. They will start to move towards the opposite pole and start the assembly of spindle fibers. Spindle fiber nyanga chimburda protein fiber and soya ko zu. And during the prophase, the nuclear membrane disintegrates. Nuclear cell nucleus chomarwe. Nucleus ki nuclear membrane kali mewa chadwe gozur. They will start dissolving. They will start disintegrating. Nuclear membrane mewa chadur. Nuclear membrane mewa chawi bana the chromosomes will be now floating in the cytoplasm. Early prophase kabdila prophase ko sudu zambala cell yore. Tinalo nucleus yore. Ani prophase to de kabla kharchi gore cell nanglo gi chromatin sama thread like structure dige sama chromosome la gyure go jure nuclear membrane mewa cha dure ani centriole dige sama opposite pole la dur cell gi opposite pole la dure ani dinanglo hekyo gi membranous andanga jo endomembrane system yo endoplasmic reticulum the Golgi apparatus dige sama mewa cha dur so these are the events which happens during prophase, so you should copy it down. Prophase kabla khar chungor lan dige sama chungor da. That this is diagrammatic manner na taya imba na if you want to see this in a diagrammatic manner. Sorry, kina zo dinde dostina digre. Ro. Mambu dela dikhare the interphase ki nucleus rada. The interphase stage larda. During interphase you can see there is a nucleus cell ginanglo nucleus to. nucleus nanglo khachido all the DNA materials they are present in the form of chromatin, a chromatin thread like structure redo ani chromatin yo masi nuclear membrane gichilo khardo centrosome ni do prophase nanglo dus enter chewada. When it enters into prophase, what happens? The chromosomes, chromatin di kharchire, chromatin they start condensing into chromosomes la gyurju do sama khaji do untangled chani condensed chani ro ani rod shape chromosome structure la gyurju do ani chilo le yo ge centrioles the centrosomes di khachi do khonsu sama pha opposite pole la do da chik north la na chik pa south la yo do opposite pole la do. And then what happens? Nuclear membrane disintegrate chagarte. Nuclear membrane starts to disintegrate. Nuclear membrane mewa chaya go chure ani centrosomes or centrioles they will start assembling spindle fibers. The napodo din so kharre la wimena spindle fibers yo and spindle fiber ring do they will start now attaching themselves to the chromosomes. Tanji chromosome form chege jo mare ta dige sama khajire nuclear membrane mewa cha do zambala now they are floating within the cytoplasm ani spindle fiber the yon khonjo gala jare ko zur so these are the events which happens during the prophase. Remove the over the then next kare the prophase ke jela metaphase chung the metaphase ki kabla all the chromosomes taji ye chromatin chromosome chadar lamba chromosome di kharcher chromosome chaya jo mare condensation process the ongoing process reda but at metaphase the chromosomes have completely condensed chromosomes dike sama complete condensed chazar ko peptide chazar and they are clearly visible under microscope. Chromosomes dike sama khajire PCO thongul ngazo microscope wala tawe mane during metaphase we can clearly see the chromosomes of the cell. Therefore it is good for karyotyping. Isi kiranzo chromosome sama matam ni par gyawo yobe mana par gya tuzu yaksho di khadure la mana when the cell is in metaphasic stage. Isi cell the metaphase stage la yobe mana kiranzo chromosome dike sama PCO thongu yoba sonza karyotype chele lo per karyotype la khare chromosome gi par lo chromosome sama ma phangdi cell chigne ma tyenirda azu subuna ni khazo yolos chromosome ship shedu yorla so chromosome ship shedu chazang co pe thongur kisi kira metaphase stage ne ma tyenjero matam ne par gyawi mana you can get a very good karyotype. You can see the chromosomes clearly. Okay, and without nuclear membrane sama mewa chazaro chromosomes they are spread in the cytoplasm na yoro cytoplasm na yama khamde yoroshto atesia chromosome la di kharig dup sha yore dinge da ngazo jangsal da nyela so cells ngazo cell ki chapter na lo la video last na ngazo khar jambe nucleus the chromosome ga jambe chromosome na lo la khari la there are two sister chromatids. Chromosome is made up of two sister chromatids. This sister chromatid chigre, this sister chromatid chigre. So these two are sister chromatids, right? And sister chromatid the call they are joined together at the centromere. Centromere there. And the centromere gichilo de la career they have got a disc-shaped protein structures called as kinetochores. They are ngomburush tongudo. Kinetochores. Kinetochores la the nenga chimbu khachin re la wemena thanji de spindle fibers yo. Spindle fiber dige sama ni chromosome la jargila so chromosome gi saja khol jargila wembena kinetochore la saja del jargir. Centromeres has small disc-shaped kinetochores which serves as a site of attachment of spindle fiber del jargir. The spindle fiber gi phajo push cheni spindle fiber starts pushing, right? Phajo sane pole ne push che jo pole ne push cheni achire they will start moving the chromosome phajo move che ko zur and then all the chromosomes they will lined at the equator. Chromosome yojo changma haji cell na lo yojo chromosome yojo dige changma they will align at a particular location called as metaphasic plate. So the chromosome move to the center of the cell and that plane is called as metaphase plate. So dikhiranzo remove dostana tigir and chromosome dige changma phajo khator deyo tel chromosome changma phajo khator dedo the spindle fiber del journey phajo theory general and chromosome dige changma hajira line signal tigir plane signal the plane dilanga zo khalegre metaphasic plate. It is right at the center of the cell penenga chomburta kisi chromosome changma central le matu wimbana the cell won't move into the next stage metaphase ki chela next stage kharin odina zugmar so the kima takta sha thoye gi lenge the switch gore spindle fiber den zugi chigor spindle fiber it pushes and pulls the chromosome and aligns all the chromosome in the midline equator ro de shagdo so the metaphase ki khejerta so The metaphase ki jela khare anaphase. Now all the chromosomes are aligned at the metaphase place. Ta kila lesa so. Kila lesa jela tsangma kharchi yore. Phajo side ki spindle fiber la del chik jar de yore. Sujo side ki spindle fiber del chik jar de yore. Kiras de seu junsa side spindle fiber dine chik yongyo del jar do. Phajo ni yongyo del jar do. So chromosome ni in the sister chromatid ni yo marwe. Ni ni ga khaji gore side ni ne pa tu de yore. Spindle fiber ki. Bas. Seu junsa side ni le tu de da sister chromatid chik phajo side tu do. Sister chromatid chik sujo side del tu do. ता दिजेला खारे नेक्स्ट दिखारे एनाफेज कब खर्ची गोरला ना ता टग ऑफ वार दोरे गाजो थगदेन सी यो दिनंग सिंग खाची गोरे सिस्टर क्रोमैटिन नि दि फासो थेंगोर दोजों से the spindle fibers they start pulling the sister chromatids and the two sister chromatids they get split at the centromere simultaneously ro thuzu chigla khachi gre ta ten da ma de ten ro thuzu chigla khachi gre sister chromatid dige sama khachi gre they will split at the centromere kozu keba dine chedu tanji centromere jar de yo ni spindle fiber gi pol ni pa ten we gen gi ro sister chromatid ni khachi gre split chedu yo chung so and each chromatids they will move towards the opposite pole chromatid ni ga khajir they will start moving towards the opposite pole now each tadil sister chromatid lag marda tadi nenga chumburda jinge la khuni nyamdo jar de zambala sister chromatid simba chromosome chigimba now when they are separate split chesa vejela when they are separated each sister chromatid is now chromosome of a future cell this sister chromatid chigre ini loga chasa vejela ta this sister chromatid lagma ko khare chromosome chig char cell shema gaji ki chromosome char now they are moving towards the opposite pole now the chosa vejela ani khare next khare telophase the telophase kablu khare chungore la wi ba na chromosomes they reach their respective poles chromosome dikhajere rakora togo sa saje jomare thendu jam phane spindle fiber ki thero opposite ni la respective pole lesser saje der imane dus pha roje khuni khaje do opposite pole le do let's have jela the chromosome they start to decondense thaji prophase kabdula condensation chowimba chromatin ne chromosome chaimba telophase kabdu kharchi gore chromosome ne ya angya khaji gore chromatin na gyur gore so individual chromosomes are no longer visible you can see chromatin materials at the two poles chromatin tongre and then there is reassembly of nuclear membrane yangya form che ko zure tinyamdo lo nucleolus ta ani Golgi complex ta endoplasmic reticulum sangma kone form che ko zur now trick lauda telophase ta prophase ni dikhare tokjo ro cha so whatever is happening during prophase opposite is happening in the telophase na opposite de chungdo prophase na lo la chromosome condense chiyo telophase na lo chromosome decondense chagdo prophase na lo nuclear membrane la du yo telophase na lo nuclear membrane gone form chigido prophase na lo organ dike sama la du yo Golgi complex endoplasmic reticulum dike sama meo cha du yo telophase na lo yangya reform chigido so you can think about telophase as a opposite of prophase is it temperature way okay so the telophase so next khare the telophase ki jela khare la win bana ani cytokinesis after all these things happen now nucleus divide chesa so now the nucleus has divided prophase metaphase anaphase telophase chenin khachire cell na lo yogyo nucleus ti ni la chasa so ta khare che ku gore cell ni le thumu ni le to go na cytoplasm the divide cha go gore cytoplasm divide cha wala cell membrane ni the divide cha go gore cell jing ni la cha go gore di khare next di khare la win bana cytokinesis cytokinesis is the division of cytoplasm or hence the division of cell koran divide chi yo do cha so in animal cells and in plant cells the cytokinesis happens in a different manner cytokinesis la di plant cell the animal cell na manda chungurda in animal cells there is a formation of a furrow in the cell membrane animal cell dina khajire ikima the intestine khajire furrow ro form chir furrow la na ta kare kimana ma deep de gyoro shmin de gambu ro chuma so na kimana kuba damsha gyoro charu and the deepening of the furrow di mama deep sar do and kuni de tu and khajire lokan ni le gyutur deepening of the furrow joins in the middle and divides the cytoplasm into two kesi gambu ro jorda imane ma thakbaji kiran kubaji karro kubadi thamro ta wa imana khajire namji ma do kuni che dura ni khajire gambu ni le gyutur so lower animal cell divide chadandi there is a formation of furrow in the cell membrane there is a deepening of furrow, and furrow joins in the middle. When the furrow joins in the middle, it divides the cytoplasm into two. That is in animal cells. The plant cell nanglola and this chair yung markajina cell wall yorda. Plant cell nanglola cell membrane chebu koran divide chawe cell wall yung marwa. In plant cells, wall formation happens at the center of the cell. Cell dinte to tushi tubiya jiyo bimbana. Tambu khajir di para de la cell wall ki material langar. Sabaya la wanashi. Cell wall ki material langar. Cell wall form chiri. Center of the cell. And cell wall ki precursor dilanga zo khalla cell plate. It starts forming a cell plate. Okay. Cell plate form che. And the cell wall the full cha sa wena nanglo cytoplasm ni the loka cha. And then it becomes two cell. And there is distribution of cell organelles. In both these cases, there is distribution of cell organelles as well. Mitochondria ke chanyumbu kopsha jegre, chloroplast in a chloroplast chanyumbu kopsha jegre ro. And Golgi apparatus ta, and endoplasmic reticulum that the sama hajire, cell touching ni likurgoro. Nigala pay would divide cheat. Nigal tata raya. Is it tata mara way by na? Cell chila mitochondria samara, cell chila mitochondria che mara way, it cannot survive. So that's the reason there is distribution of cell organelles into the two daughter cells. Okay, and then you see there is a formation of multinucleate cells if the karyokinesis is not followed by cytokinesis. You see cytokinesis machunga chewing by na. Karyokinesis chungsar the cell download your nucleus thingy the chasar. But this jela cytokinesis machunga by na, that will result in the formation of multinucleate cells. Cell ching now nucleus mangbo yoyo form chigre. That is called as synthesizes. Multinucleate cell namazo khalagre synthesizes, which means it contains two or more nucleuses. Nucleus manga yoyo or nuclei. The kharchin chigore karyokinesis chung cytokinesis machung wa imba na multinucleate cells ka condition dekhi. Okay, I hope it is clear now. The thing azu khar gan jang law imba na. Abu karyokinesis che ani cytokinesis kala. This is the formation of, this is the cytokinesis in animal cell. The animal cell nalo cytokinesis totan diche dikhare plant cell nala cytokinesis totan di dina khachido animal cell nalo nucleus divide chasa vejela yela furrow form chi the furrow form chi go jumje khare protein ring form chi microfilament ring the tight sar do sambala there is formation of furrow, cleavage furrow the tight sar do sa vejela khajire cell tumbuni la matang shir. Animals plant cell nalo khachi there is a formation of cell plate form chi da gimala cell wall ki precursor cell wall form chi da deep cell wall form che ki material dige samo suite gore endoplasmic reticulum the Golgi apparatus dige samo suite gore there cell wall form che za vejela cell membrane saw form chire and then the plant cell will divide into two cells. Cytokinesis ki divide khewani de yabu ha ko achima animal cell nalo da plant cell nalo la cytokinesis ni de loga yorda. Okay, iche eta di remu de yangya ro tete do kiran remu do chigla tishan ri tende ro tiyo imene. If I ask you questions, right, if I ask you a question like differentiate cytokinesis between animal cell and plant cell, you can draw this diagram. Cell plate form chido inalo khardo cleavage furrow form chido. Okay, so that's the difference. And the entire process of the cell cycle. Interphase, prophase, metaphase, the pro-metaphase and the jangmar. Prophase, the pro-metaphase and the prophase is the early prophase. Pro-metaphase is the late prophase. Early prophase, late prophase. Late prophase ne khajire metaphase. Metaphase ki jela anaphase ki jela telophase ki jela cytokinesis cheni cell nila. So this is a entire cell cycle. Okay. So I hope you are clear regarding this one. Okay, now finally we will talk about the significance of mitosis. The mitosis ki significance kare ye khala sotanji cell cycle and cell division na mazo ki khamanda ni kan jangal so mitosis and meiosis kan jangal so. So we have so far discussed about mitosis, anti-mitosis process kan jangsar da. The mitosis ki significance kare, what is the necessity of mitosis, what is the function of mitosis? Mitosis usually occurs in diploid somatic cells, with exceptions in lower plants and some social insects where haploid cells also undergo mitosis. Mitosis it happens in diploid cells or somatic cells, body cell. Mitosis occurs in diploid for example chromosome shift schedule yo 23 pairs yo chromosome yo den cell la ngazu khala gre diploid cell la wa somatic la mena ngazu subu gi cell wa diploid cell na chung go res somatic cell na ta di ngazu subu na lo but in lower plants and in some insects mitosis can haploid cells can also undergo mitosis. Haploid la example khala gore if there are only 23 chromosomes, not 23 pairs of chromosomes, just 23 set chig mo me wi bana di la ngazu khala gre haploid cell la ngazu subu na lo chromosome 23 pairs yo la ba so diploid number dikha re 46 cha so 23 pairs lo example 46 ra so diploid cha do ngazu cell di kisi cell chig na lo la 23 pairs me wache 23 mo me wi bana only 23 set chig mo me wi bana this cell dikhar cha do haploid cell cha do ngazu gu subu na lo la haploid cell di mitosis do marta it won't undergo mitosis, it will always undergo meiosis so mitosis like the sugi chingore diploid cells in sugi so there is only this exception, lower plants and some social insects, they in them haploid cells can undergo mitosis. The significance ke je wi bai na answer simple kharche hai mitosis ki ta significance khare why is mitosis necessary for our body. It is simple answer na longas khajore it helps in growth and repair. Ye tanji la so ngaju sang thobung ching ne tup sha gyor das ngaju sang thobung ching ne chung gyor da la so zygote ne zygote divide che ne ngaju multicellular organism charwa so multicellular organism chaya la zygote divide che go da divide khare ge through che go re mitosis process ke through che go so mitosis is important for growth ngaju chinju gabla khandi ta thanda chimbu cha wora this chare ngaju chimbu through cell division ki process dikhare mitosis ke process through our body cells, somatic cells, they divided through mitosis. Therefore, we have grown into larger organism from babies, from baby neta adult charwa. Then repair la example khalo wore. Whenever some cells die, pension ma do so bi mena cell chigoro. The cell dikhaji wore, replace chigoro, repair chigoro. The repair dikhari through chigoro bi mena mitosis. Our skin cells will undergo mitosis to repair the lost skin cells. Nima takba ngazu skin share chigor, skin cell chigor da. Ine khachi gor ngazu pakwa ngazu mewa cha rumda. Jido pakwa ane sawa ke yungor. Iki sawa di khandis ke gor la na the skin cells they undergo mitosis. So it is important for repair. So it's necessary. So you can look at regeneration process. In some animals there is something called regeneration. Simjeng kashe na la, soja kashe na la. Isi ne lapa do tup shugu do tup chewe mena. Lapa sawa ke yong shugu sawa ke yong chigdo. Regeneration process kana chiklo da. And you will understand mitosis is important over there as well. Mitosis play a role in that particular organism as well in regeneration process. Okay. Also another simple answer kharla wore mitosis is important for growth and repair. Is it clear? Okay. So with this we have come to the end of the chapter. In the next, not the end of the chapter, at the end of this particular topic called mitosis, in the next topic we will discuss about meiosis. Meiosis kana jang. So thanda thari ngazu dina khal jare mitosis kan jare next meiosis. Okay, so read about meiosis. The meiosis is this complicated tha. Mitosis is very simple, right? Meiosis is little bit complicated, but it is there is some similarity between mitosis and meiosis, right? But meiosis is little bit complicated because it happens in 2 steps. And there are many other processes involved in that one as well. So please read about meiosis before you watch the next video. Okay, thank you.